Welcome to the FAQ Shack. Today we are tackling a really fascinating and honestly kind of mind-bending topic. This one comes to us from a listener named Alex, who asked a question that gets right to the heart of what we even consider to be an infectious agent. And hey, if you've got a burning question you want us to tackle, drop it in the comments below. We absolutely love digging into these things.
Okay, Alex's question is perfect. Is it a virus, a bacteria, something else? Well, let me tell you, the answer is something else entirely. And it's one of the most surprising and frankly weirdest discoveries in all of modern biology. It really forces us to rethink the very rules of life.
So let's just get right to it. A prion isn't alive. It's not a living organism at all. It is, simply put, a protein. But it's a rogue protein. A protein that's gone bad.
Now, the name itself, Prion, which was coined back in 1982 by Stanley Prusiner, is actually a huge clue. It's a mashup of Proteinaceous Infectious Particle, and that was a radical idea. The keyword here is proteinaceous. It suggests that you can have an infectious agent made only of protein. No DNA, no RNA, none of the stuff we thought was required for infection.
And this slide right here really shows why that idea was so nuts. Think about every other bad guy we know in biology. Viruses, bacteria, fungi. They all have a genetic blueprint, right? DNA or RNA that tells them how to make more of themselves. Prions? Nope. They have absolutely zero genetic material. They're just a corrupted version of a protein that's already in our bodies. And that's what's so sneaky about them. Because it's technically one of our own proteins, just in the wrong shape, our immune system doesn't see it. It just flies completely under the radar.
So that leads to the million-dollar question, right? If it has no instructions, no DNA to copy, how in the world does it multiply? Well, the simple answer is, it doesn't. Not in the traditional sense. Instead, it kicks off this terrifying and incredibly efficient chain reaction. Think of it like this. You've got this one bad guy prion, the misfolded one. Scientists call it PrPSc. And it bumps into a perfectly healthy, normal version of the same protein, which is called PrPC. The normal one is all flexible and coiled up. But when the bad one binds to it, it forces the healthy protein to snap and refold into its own corrupted, rigid shape. And that change is permanent. So now you have two bad proteins. And what do? They go off and find two more healthy ones and convert them. Then it's 4, then 8, then 16. It's a runaway chain reaction, a domino effect on a molecular level. And as this cascade continues, these misfolded proteins start sticking together, forming these nasty clumps or aggregates in the brain. Over time, these clumps are toxic. They kill off brain cells, they kill off neurons, and they literally leave microscopic sponge-like holes in the brain tissue. It's this physical damage that leads to the awful and always fatal symptoms of prion diseases.
So you have to wonder, where does that first domino come from? How does this whole nightmare even begin? Well, in humans, these diseases can be traced back to one of 3 different starting points.
And what's really unsettling, if you look at this chart, is that the vast majority of cases, we're talking about 85%, are what we call sporadic. That's a scientific way of saying they seem to happen for no reason at all, just completely at random. The current thinking is that it's just a case of incredibly bad luck, where one single normal protein spontaneously misfolds and kicks the whole thing off. And this table breaks it down nicely. You've got sporadic, which is that random, spontaneous event. Then you have genetic cases, which make up about 10 to 15%. This is where a person inherits a gene mutation that just makes their protein a little less stable, a little more likely to misfold. And then there's that tiny slice of the pie, about 1%, called acquired. And this is where the infamous mad cow disease fits in. It's when a person is exposed to prions from an outside source, like from contaminated food or, in some very rare cases, medical procedures.
Okay, this all leads perfectly into another fantastic question we got. This one's from a listener named Maria, and she really hits on what makes these things so uniquely scary.
That is an excellent, excellent question. I mean, we cook our food to kill bacteria, right? We use sterilizers to kill viruses. Why doesn't that work on prions? Well, the answer goes right back to that corrupted, misfolded structure we were just talking about.
So that ridge's shape isn't just for show, it's like biological armor. It is incredibly stable. So stable that prions can survive crazy amounts of heat, radiation, and even harsh chemicals like formaldehyde that would totally destroy a virus or a bacterium. Even our own body's internal cleanup crew, these enzymes called proteases that are designed to chew up and recycle old proteins, they can't break it down. The prion just resists them. And this near indestructibility is why they are such a nightmare in healthcare settings, because they can survive standard sterilization and persist on things like surgical instruments.
So for decades, this incredible toughness made diagnosing a prion disease in a living person pretty much impossible. Because the rogue protein is so similar to the normal one, and you're looking for tiny, tiny amounts in the beginning, the only way to be 100% sure was a brain autopsy after the patient had already passed away. So imagine the challenge. How do you find a single bad protein, a needle in a haystack of millions and millions of perfectly normal proteins, and find it early enough to actually do something about it? For a long time, it seemed hopeless. But then, a truly revolutionary test came along called RT-Quick, and it changed everything.
The absolute genius of the RT-Quick test is that it basically uses the prion's own deadly superpower against itself. So, scientists take a tiny bit of a patient's spinal fluid and they mix it in a test tube with a whole bunch of healthy, normal prion protein that they made in the lab. Then they shake it really, really fast, they quake it, which is where the name comes from. And if there is even one single rogue prion in that patient's sample, it will start that domino cascade, converting all of that lab-made protein. A special dye in the mix lights up when this happens, so you get this bright glowing signal. It literally amplifies that single bad protein into a billion, making it super easy to detect.
And how reliable is it? It's incredible. We're talking 99% or higher specificity for the most common human prion disease. It's so good that it's now officially part of the diagnostic criteria. For the first time ever, doctors can give a family a definitive diagnosis while the patient is still alive.
And being able to diagnose early cracks opened the door for the most important thing, treatment. You see, the whole disease process needs that normal healthy protein, the PrPC, to keep going. So the logic is simple. What if you could just remove the fuel for the fire? And that's exactly what new experimental therapies are trying to do. They are developing genetic treatments designed to tell the body to simply produce less of that normal prion protein, hopefully slowing or even stopping the disease in its tracks.
And this brings us to the final and maybe the most hopeful question of all. This whole mechanism of a misfolded protein triggering a chain reaction, well, it turns out it might not be unique to prion diseases. Scientists now see similar patterns in other devastating neurodegenerative diseases like Alzheimer's, Parkinson's, even ALS. So the big question is, could cracking the code of this one bizarre rogue protein give us the key to understanding and maybe even treating a whole range of brain disorders? It's a huge, tantalizing possibility. So thank you so much for that amazing question, Alex, and thank you all for joining us here on the FAQ Shack.